So I've been in the game a long while. This is me penguin wrangling in 1988. I've been really lucky as a biologist to be in. I've been to the Antarctic. I've been to the Arctic. As Glenn said, I've been getting these lifetime achievement awards, which is awesome, and I'm very flattered. But it also means clearly it's time to go. That's what usually happens here. And so what I'm going to do is, let's say, review my career. Let's see if this one works. Yeah, okay. So basically, I've been in the game about 40 years now, which is frightening, really is. I can remember it wasn't that long ago when I thought, I'm a new member of the faculty here at SFU, you know, I'm not anymore. So I have basically taken a slow 40-year walk through the avian reproductive cycle. And I'm going to use this as a framework to describe to you some of the work we've done. And I'm going to start with egg size, because that's actually my first insert back in 1994, and then we'll get to some of the others. This works. Yeah. So through my whole career, I've worked pretty much, most of this stuff has been done on two systems. This European starling, which we have in nest boxes. Actually, I saw starlings out there singing away. They're singing away at the moment. And then zebra finches in the lab. And my first lesson here would be develop a lab study system as well as a field system. So what I've done for the whole of my career is test ideas in zebra finches where it's tractable, and then try and take those ideas out into the field with the nest box breeding starlings. Aware that not everybody, well, perhaps everybody in this room is a bird person, I'm gonna, oh, you don't see that, that's good, I can ignore it. So what's the novel perspective? Lots of people do avian reproduction. As Glenn said, we really tried to focus on female reproductive traits, and I'll get into that a little bit. I really focused on individual variation. I have sometimes published comparative analyses, but that's not what I'm interested in. I want to look at variation within a population or among individuals. And then particularly I'm interested in physiological mechanisms. And the framework has been an evolutionary one, a life history one. My funding has been from the Ecology and Evolution group in NSERC the whole time. I start my proposal by life history traits and life history variation and trade-offs and things like that. But then I get down into the weeds and do the physiological mechanism. I'm going to mention briefly a few other species because I actually think that some of the findings we've had are kind of can be generalized across taxa, and that hasn't been picked up at all, it turns out. And so I'll get into a few examples of other things too. So let's start with egg size. Why did I settle on egg size in 1994? When you're a new faculty member, you think, what the hell am I going to do? I need a research program. And I'd done a lot of work, and I thought, well, clutch size, no, that's been done to death, and I'll get back to that one. And then I thought, well, egg size is pretty cool. And why is it cool? Firstly, it's highly variable. It's a life history trait. Everybody acknowledges that. So there's that evolutionary perspective. It's massively variable. So this is from a review one my PhD student did, Julian. And what that top graph shows is that within a population, some females are laying eggs half as big, 50% bigger than the smallest eggs. And it's not uncommon for that to be twice the size. So within a single population, some individuals producing eggs that are twice the size of other females. And the bottom graph shows that most of that is individual variation. It comes from among clutches. It doesn't come from within clutches. And so we have a trait which is, we have a trait where there's a huge amount of variation, which is what I love. I love explaining that. It has high repeatability. These are a couple of early graphs that we did looking at taking multiple measurements from the same individual. If you're going to do individual variation, you have to have a system where you can take multiple measurements. Otherwise, there's no point. And so you can see repeatability. The general idea is that there's repeatability. Julian argued there's no plasticity or limited plasticity. We're actually revisiting that a bit now because, as you can see, there's some deviation from that line there. And that could be genuine plasticity. And it would be interesting to tackle that. So as I said, my first insert was basically what are the causes and consequences of individual variation in egg size. And when I started this in '94, you would have, if you'd read the literature widely, which I'm sure you all do, and I'll come back to that too, you would have thought this problem had been solved. So here's Tim Cluttenbrock's classic book, which I really like, Parental Care in 1991. In birds, positive relationships between egg size and chick survival are common. And then another example, positive correlations have been found in most species studied. The problem is solved. Large eggs are large because they produce a fitness advantage. Okay? And that's fine. It doesn't explain the physiology, which I'll get to. But then if you actually started looking at that stuff critically, and again, very rarely have I been the first person to say any of this, in fairness. So there's this paper by Muller pointing out that, in fact, there was a confound in all of these studies, which was parental quality. So the simple idea is a good quality female produces large eggs, but she also feeds those chicks well, and you can't uncouple a direct effect of egg size. And so I wrote the review basically saying that, and concluding that actually, even though there were 50 studies that I found in this review, there was little unequivocal evidence to date to support this. And so this became, to me, a valuable question. Why lay large eggs? The question actually should be, why don't all females lay the smallest minimum viable egg size? And I'll show you why that is, because we still fail to identify any fitness advantage of large egg size. And it comes at a cost of your total fecundity. And so it would make much more sense for this to be the strategy that should be pursued. And that is a good example of a massive question that we haven't managed to resolve. I failed to resolve too.
Excuse me.
Yes.
On Zoom, we just can't see the slides advancing. We're still on this first slide.
Oh dear.
Okay. Let me try and fix that.
See what I said? We had the same problem at SFU, and our seminar organizer doesn't believe in hybrid. They just want to get everybody back in the room. And of course, I said, well, we've got to be a bit more aware than that, because I'm going to get it in the net otherwise. But every week is an adventure.
It really is, and I have no idea why that is, but it's consistent, at least that is. Back here.
Well, maybe I'll. Shall I carry on talking at least?
If you want to, yeah.
So when I got back to starting to give talks like this one, and I'm giving a talk in another meeting, and I gave a talk to a grad seminar last night, I thought, what am I going to talk about? Like COVID, I've done that. And then I thought, well, that's really sad. But then, well, if you review it with a positive spin, then that's maybe not so bad. So that's where we're going with this. And so I started to catch up on exercise, because I haven't really worked on exercise explicitly for about 10 years now. And I thought, well, maybe someone has run with this and solved everything, and that would be cool. I doubt it. And so I started to look at recent studies on exercise. And here's an example. This is a paper that claims that there is a relationship between egg size and temperature, but there's a confound there, or an association with helpers in the nest. So this is a cooperative breeding system. And their argument, and the paper was that females are adjusting egg size systematically with temperature, but only when they have helpers and not when they don't have helpers, which could be really cool. And that's okay. But the way I look at this is that there's a real conceptual conundrum here. This is the line. Can you see that now? Okay. I'll get Gabby up there. But anyway, that's fine. I'll roll with it.
Let me just hide the bar, and you can be.
So I'm trying not to be negative about these studies, but to me, they miss the problem of individual variation. So what I've put in the circle there is perfectly good females, individual females, that are laying even massively large eggs and even smaller eggs. Yeah. So their argument is that an adjustment of about 50, whatever that is, 50 millimeters of volume on an egg size of 1300 is adaptive. And I'm going, well, how come, if that gives individuals an advantage, what are those females doing at the top? How come they're not the super females that are really kicking the butt of everybody else? And similarly, why aren't there loser females at the bottom? And so there's been a lot of focus on plasticity, still on plasticity, but it's that little wiggle room around the big line I showed you, the repeatability line. It doesn't explain individual variation to me. And here's my, oh no, this isn't working. Okay, so what I was going to show you, you could play with that again. So what I was going to show you was an insect example, because I like to look at other taxa, and it turns out in the insect example I'm going to show it's exactly the same. There's basically a cloud here. Hit the advanced. What are you doing? I'm not doing it. Oh, okay, so just to show you this is a common problem, the paper focused on that one line there, which is one population. And even for that population, there's quite a lot of variation and unexplained variation. And yet there's these other populations which have this massive overall variation for the species, individual variation, and they didn't even trouble themselves about that. So that worries me a little bit, that we're missing the main questions. Here you go, hands on. Okay, so there have been a few good studies recently. This guy, Chris, who you can just about see there, has been doing really good work. And this is a very large field experiment in pied flycatchers, which showed that egg size can have a fitness advantage under very extreme bad conditions. So if you look at postnatal condition, there's a little bit of circularity here, but when things get tough enough, egg size matters, okay? But the question is that enough to drive selection? How often does that have to happen? And he then has done a much better job of me. He actually criticized my 1994 for being narrative, which is true. I didn't do a meta-analysis or anything, so he did a meta-analysis. And if you look down here, we still have absolutely no evidence for egg size in birds having any long-term fitness consequence in terms of the things that are fundamental: recruitment or lifetime reproductive success. So the approach we took to this, and this is where we get into this conceptualizing thing, is I wanted to know what the physiological basis of egg size variation was. And I thought if I could do that, I could then develop tools which would allow me to manipulate egg size and then directly show that egg size had fitness consequences. Actually directly manipulate egg size in individual females. I failed for reasons which we'll get into, and it's kind of fascinating why I failed. But I actually thought I had a good system because we understood the physiology of egg production from all the chicken work that had been done. And so here's a very simple cascade. We have a hormonal signal, which is estradiol, goes up during the breeding season, drives all of the egg production system that Glenn was talking about, primarily stimulates yolk precursors. There are two: vitellogenin, which is largely protein, and then VLDL, which is lipoprotein. I'll come back to that. And then between those, the idea was my idea was that they would dictate egg size. So if you have a big hormonal signal, lots of vitellogenin, you produce a large egg. Pretty simple. We started doing this, and I got a bit famous for this, showing these at meetings. These are shotgun blasts. So here's vitellogenin against estradiol. I was predicting they'd be tightly correlated or positively correlated. Here's follicle mass, so egg size basically in terms of plasma vitellogenin. Again, they're shotgun blasts. What I want you to note is the amount of variation there. It's not that there's no variation, there's massive variation. And this was really troubling because estradiol, for example, is a very potent hormone and it has negative effects. It decreases red blood cell production. It just, we live in a world where natural selection is not meant to generate this. It's meant to generate some sort of fit between costs and benefits. And I thought estradiol, you shouldn't have too much of that slopping around. It's very bad for you. So here's another fundamental question which hasn't been answered. Why is trait variation, in this case egg size, but I think you could extend this to most life history traits, and I'll come back to clutch size in a minute, back time. Why is that independent of underlying physiological traits? Now, we persisted a little bit, and I can give you a better answer. I think that yolk mass is dependent on a couple of things. Rate of yolk precursor uptake. So that's that last step there. The vitellogenin's in the circulation. It has to be taken up into the developing follicle. That's a receptor-mediated step. It seems that that's critical. And then the thing you can't see here is it's actually related to receptor mRNA for the vitellogenin receptor. And that's this graph here. So I think I can tell you, what physiology underpins a large egg size, but it's not the answer I was expecting. And one of the things is receptor uptake, but I think this lipoprotein metabolism is another one. And what's fundamental here is, so you're all producing VLDL at the moment in your bodies. It's what we call generic VLDL. It's the stuff you use to fuel the activities you're doing. But in a female bird and in a female human, you switch to producing what's called a yolk-targeted, at least in birds, yolk-targeted VLDL. It's a different type of VLDL. And the theory is the female can't use that for her own needs. And so I think personally, this switch to this yolk-targeted VLDL is key. And we've got, perhaps I'll fast forward through this, but the backstory here is, I'm going to get to the male bias in a minute. Why have we got a male bias in reproduction? When you bring male birds into captivity and you shine lights on them, so you use artificial photoperiod for long day lengths, it turns out you can fool them entirely. They'll develop their gonads, they'll produce testosterone, they produce sperm, they're almost certainly able to fertilize females. When you bring females into captivity, they don't do anything. Doesn't matter what you do to them, by and large, they don't do anything. And so that's why we've got this male bias, because males were easy to work on and females weren't easy to work on. And I'll come back to that. But basically what I've done here is push some females as far as I can. So I've got a long day and short day treatment here. So that's like, fall conditions and spring-like conditions. And then I also shot them up with estradiol, which is the hormone I think is important. And what the top graph shows is that you can get a vitellogenin response. So vitellogenin, you can fool females into expressing vitellogenin in captivity, but the VLDL is just on the ground regardless of the conditions. So this is why one of the reasons I think VLDL is critical. And we've accumulated a bunch of other, yeah. So, no, it's not working out. Every time you mess with it, man. Yeah. There we go.
You just have to click.
Oh, clicking. Okay, good. I'm going to get a solution. Yeah, so it's not too much detail, but over years we've accumulated a bunch of other evidence that VLDL itself, this yolk-targeted VLDL, is really critical. It responds to natural fasting, it responds to stress, all the things you think might be involved in cost of reproduction and stuff. And it actually predicts egg laying ability. So that's something I'll come back to. If you try and force females to lay more eggs, the ones with high VLDL can do that better. And so I really think this is, if we get to what is the physiological basis of fundamental trade-offs, in this case, between investment in eggs and self-maintenance, which is a pretty fundamental trade-off, then I think lipoprotein metabolism is a key. And because this is a marine department, look at this, sea urchins. I came across this example. And so these are two species of sea urchins. And one of them has the ancestral mode, which is relatively small eggs. And the other one has evolved large eggs. They're not to scale for the beast, but they're to scale to each other. And so, I'm fascinated by this. And it turns out, if you ask what's fundamental about this, what physiological mechanism mediates this transition, it looks like it's lipoprotein metabolism again. So they have an ancestral vitellogenin component, which is what I showed you we can switch on fairly easily in birds. But then this species here has evolved this secondary deposition of lipids and other non-vitelogenic proteins. That's that lipoprotein metabolism part. So I really think, again, we've kind of highlighted the fact that lipoprotein metabolism could be really cool and central to a lot of these trade-offs, and nobody's really picking up and running with that idea. Yeah, so then the idea is, once I know a little bit about the physiology, I can take that out and try and use it. And I'm going to summarize some of this, but basically, we know estradiol is important, estrogens are important, so why not block that? It's fairly easy to do. We use a chemical called tamoxifen, which is actually the breast cancer drug. And it decreases vitellogenin really successfully, so that's great. It decreased egg size only by about 8%. So there's that slop in the system again, which is really weird and hard to explain. When we did it the first time, a decrease of egg size of 8% had no effect on hatching, or had a small effect on hatching success, but no effect on offspring development. So that suggests that actually, it's hard to show there's a fitness consequence. We came back and did it again with a higher dose, and then we got a massive effect. But you have to decrease egg size by about 18% to have a really significant effect. And so here's the data. The top graph shows you egg mass by laying sequence, and you can see in our tamoxifen-treated birds, they laid much smaller eggs. And then we had this fitness effect. Someone's going to ask me, how do I know that's not just a toxic effect of tamoxifen? And I'll address that afterwards, because I'm running behind. Yeah, so then, ideally, validate it in the lab, take it out into the field, and this is where I failed, and this is where I'd like some help. So we tried this in starlings. I wanted to get tamoxifen into starlings. With the zebra finches, I can catch them every day and inject them, and they don't care. They just carry on laying. Fantastic model system. You can't do that with starlings. You catch them once at 1 egg stage, and they're just desert. At the 3 egg stage, they're fine, but it's too late then. And so we tried to use osmotic mini pumps, and you can't get enough tamoxifen in an osmotic mini pump for this to work. We did the opposite result, where we actually injected estradiol. And again, it kind of confirms the previous stuff I told you, that if you inject starlings with estradiol, you get a massive increase, almost 50% increase in vitellogenin, but it doesn't translate into any effect on egg mass, which again suggests there's this rate of uptake limitation. You can't overload the system. So if anybody knows of a better technology or a more potent anti-estrogen, let me know after the meeting, because this can be my answer, hopefully, if I can find a way to do this, because I need to revisit this and solve it. Okay, so I'm going to skip through some of this. I took this out in the version that was on my laptop, but we switched to my thing. So this is a general point that, we don't understand the physiological basis of life histories at all. Even though people have been talking about that for 20 years or 30 years, there aren't many good examples where we have the mechanism of that. What's weird here is I think we're closest in terms of egg size, but we're left with this kind of receptor-mediated process as potentially the target for selection. And yet we talk about resource or condition dependence, we talk about nutritional constraint, we talk about resource allocation trade-offs, which I think are missing the point of the mechanism I've kept. So, I've been pushing this idea that maybe there are physiological incompatibilities between systems. So here's a big knowledge gap. So now I'm going to switch to the timing of breeding one, and this is a win. So the last one is probably a moderate fail, I would say. This, I think, is one area where we finally, as Glenn said, I think we're finally getting some traction here in convincing people that females are not males. And that sounds very bizarre, but I'll explain why that is. So for 50 years since John Wingfield's early work, for example, with field endocrinology, we've had a model of timing of seasonal breeding from a mechanistic point of view. And the model's kind of captured here, but photoperiod was the driver of that. As day length starts to increase in spring, birds, and we meant by that males, but we translated that to females as well, which was a mistake, basically start responding to increasing day length. Gonads grow gradually, and then they reach maturity around the time of egg laying. Okay. There was some suggestion that other factors like temperature and food and social factors played a role in that, but they were poo-pooed because they didn't kick in early enough. Yeah, so they couldn't be predictive. But that was because they were thinking about males. Yeah. And so we have this 50-year-old model for males with this initial predictive information photoperiod with supplemental cues. The supplemental cues, ironically, were argued to be fine-tuning cues. So the female has this gradual gonadal maturation. And then just before laying, she says, oh, it's a cold year or a warm year. I'm going to use these other things to fine-tune. So they did bring females in at that point, but nobody studied that at all. Yeah, and certainly not in the lab, because you can't do this in the lab. The females don't behave properly. So we started collecting some actual data on this, and this is from our early work, just looking at how, again, vitellogen and estradiol, the fundamental players in this, change throughout the laying cycle. So this is a non-breeder. This is basically the day before we see the very first follicle. Then they develop a hierarchy of follicles, 1 to 2, 3 to 4, 5 to 6, and then they start ovulating them, so the follicle number decreases. And then this is clutch condition. And there's some interesting results there in general, but the key thing is this thing here. Non-breeders only 3 weeks before laying, and even the day before the first follicles appear at all, there's absolutely no reproductive activity in the female reproductive system. Totally at odds with that kind of model there. And so this is actually what the world looks like. Here's the male on the left, and this is data from my PhD supervisor, Alistair Dawson, consistent with this idea of a gradual effect of long day length and a gradual ramping up of testicular development. It takes about 6 to 8 weeks in a male to go from 0 to full testis function. Although rather bizarrely, they seem to be functional in terms of sperm, that little asterisk, way before they reach maximum size. And then here's our data for females with oviduct and follicle size. Basically nothing, bam, this female switch on. Why is that important? Because all of us are looking for the signal that controls timing of breeding. And because we've had a male focus, we've been looking down the line, and we've only been looking at photoperiod. And both of those things are bad mistakes. Basically, I'm going to walk away briefly. Basically, this is where we should be looking for females. And this actually brings in the social factors and the temperature and things, because if those are fine-tuning cues, this is when they're operating on the female. So because we didn't have the female focus, we've been missing the ball of where to look. And it turns out that females can switch on really quickly. This is oviduct development, plotted on that same scale of follicle development and then ovulation. And you can see, interestingly, the oviduct doesn't actually reach full size until like quite late in laying, probably because this is costly and they don't want to maintain that material for a long while. Whereas for males, testis, who cares? Just have them big all the time. And then if you look at vitellogenin again, they can crank this up really, really quick. You get this very rapid upregulation of vitellogenesis to maximum size. So females are totally capable of leaving it till the last minute, making that decision at the last minute, I think integrating a whole bunch of information, because reproduction really is costly for them. I'll come back to that one in a minute. And so here's the win. This is kind of the standard thing that I do in my endocrinology course. There's the central nervous system integrating all of this information on social and temperature and day length, signals to the anterior pituitary, signals to the testis or the ovary. And what we have in males is this sort of upstream control model where long day length is necessary and sufficient for full gonadal maturation and full gonadal function. But what we have in females now is something very different. We have what we call downstream control, where day length is necessary and sufficient, is necessary but not sufficient. So without doubt, the female switches on at the hypothalamus and pituitary. We've got evidence for that. But there is a block at the level of the ovary and probably at the level of the liver, I believe, because of that lipoprotein metabolism thing, where the females are actually making decisions at that stage. And that allows them to integrate all of this information. And we have a bunch of information that supports that. Plasma LH and FSH actually do increase. If you take a female starling in captivity and expose her to long day length, she won't grow her ovaries, but it turns out LH and the hypothalamus switch on in exactly the same way as the males. So it's not like they're ignoring that photoperiodic signal, they're just filtering it lower down and saying, well, that's necessary, but not sufficient. And then, as I said, we now know that LH and FSH receptor mediated competence of the ovary is important. So gonadotrophins are circulating in the bloodstream, but the female's ovary just isn't paying attention because it hasn't expressed the receptors. So a little bit of traction here on a few labs working on this. The sad part is this. This is from a paper I published with a group, with Marcel Visser's group in Holland. They threw the basically the kitchen sink at this. So they did genomics, they did epigenetics, they did everything, millions and millions of euros. And this was our conclusion. Well, one conclusion was that by and large it's consistent with this female-based model. But the other conclusion was we don't know what the exact downstream mechanisms are. Because we have spent 50 years not looking at them, we have no idea. So if you want a big problem to solve, then go after that one. Okay, we're good. Okay, I'm going to change gears a little bit and talk about clutch size now. So when I got into the field, I had spent some time in waterfowl biology. I worked on snow geese up in the Arctic, and waterfowl biologists are obsessed with clutch size. And so I read all this literature and I thought, yeah, there's no questions left to answer about clutch size. It's been done. But if we look at the variation, it's really cool again. So it's another life history trait that shows systematic variation. So on the top there, there's seasonal declines in birds which only make one breeding attempt a year. In the bottom, there's some plasticity. This is from one of our studies on song sparrows, where we have the standard for species that lay multiple broods. You have a standard kind of curve like this, where they lay smaller clutches early on, they optimize clutch size in the middle, and then they lay smaller clutches at the end. But you can flip that around to a seasonal breeder type thing if you mess with it. And so that was what we did. So fascinating plasticity and variation. This is data from our starlings showing that starlings appear to adjust clutch size even over 3 or 4 days. Highly synchronous, my starlings. And so everybody's starlings. And yet you seem to see some adjustment. So if we're thinking about a mechanism, then that becomes kind of important. And again, in contrast to egg size, we know that clutch size has strong fitness consequences. Lots and lots of studies showing that it really matters how many eggs you lay. Quantity is a driver of lifetime reproduction success. So then I started thinking about mechanism, and I thought, well, okay, this is great. So surely we know the mechanism of this. And nobody had looked at the mechanism of clutch size determination. So this is the key to me. And started reading literature. And once again, people had thought about it, a couple of people, back in 1990. This is work by Theo Mayer's group in Germany. Basically, common ground here, the idea that there's some sort of inhibitory signal. So females develop their ovaries, they start developing follicles, they keep marching along, 1 follicle, 2 follicles, 3 follicles, and at some point a gate comes down and says, stop developing follicles. So that's this inhibitory signal here. So here's our first follicle producing first egg, second follicle. And both of these kind of had the same idea that there was some threshold for follicle absorption. And both of them implicated prolactin. So we've got a hormonal mechanism here. Technically, I'm a trained endocrinologist. So whenever I see an endocrine mechanism, I think that's cool. So what do these have in common? Both models predict regulatory roles of prolactin, so prolactin-mediated mechanism. And the idea is prolactin is antigonadal. So it's going to shut down the gonad in terms of, for example, gonadotropin. So then you say, okay, that's cool. It's this 1990. And I started thinking about this in the early 2000s, 15 years ago by. I thought, well, surely someone's picked this up and run with it. This is such a fantastic peach to grab and solve. Surely someone's done it. And the answer was no. So this is Keith Stockman's work in the kestrel. At least he did stuff in the real world. But this is kind of the best data we had, 16 years on from the mechanism being proposed. It just blows me away that nobody cares about identifying mechanism. And you can see it's not great here. So he's got the left-hand side is a correlation just with circulating prolactin. That's probably pretty weak anyway. But then he actually injected prolactin into the birds and wasn't able to shift clutch size. So we decided to take this on, and I'm going to show you bad data first. So this is our correlational approach. So we first said, okay, we'll take our zebra finches. We'll just look at circulating prolactin and see if it relates to clutch size, and it doesn't. There's a few weird outliers here. Sometimes these birds individuals just carry on laying, which is very weird. But then we took a better approach. And here's the next one that I want to say. This is my conceptual approach. I think there's a lot of power in what I'm going to tell you about, but very few people are running with it, which is a bit disappointing. So we don't know about prolactin, so let's do it. So I talk about physiological reaction norms. So when I gave this talk last week to my grad class, I said, so someone tell me what a reaction norm is. And there was stunned silence, which is really demoralizing. And I will come back to that point too. And so I'm sure you know what a reaction norm is, but basically here's an example. So to generate a reaction norm, you need multiple measurements from the same individual again. So we're back to this. If you're going to do individual variation, you have to commit to measuring the same thing multiple times. This is the relationship between laying date and temperature. This is actually from Great Tits, I think. And so what they've plotted here is individual slopes from like 4 or 5 different years where they have both laying date and temperature for different individual females. And the nice thing about reaction norms is it generates different statistics than just a single point estimate. You can generate the slope, which is the relationship between the trait and the environment for each individual. And you can see individual 1 is very responsive, appears very responsive to temperature, whereas individual 4 seems less responsive to temperature. And then you can do, on an average environment, you can look at the elevation. And so these were ways of characterizing and utilizing individual variation. And so we thought, could we do this in a physiological perspective? And here's one example that I used in a review on this. This is a little bit complicated, but I told you earlier that one thing estradiol does is shut down red blood cell production. So if you have a high estradiol during laying, those birds become anemic. And so what we've got on the y-axis here is basically the change in anemia. So these individuals are very anemic, the other one's less anemic. And then I measured that once for baseline estradiol signal, and then I decreased the estradiol signal using tamoxifen that I did before. And so basically I've got a reaction norm here of 2 different environments, but these are physiological environments. There is physiological variation. And I don't see why this isn't a valuable approach. And you can see again, very interestingly, that this individual here seems very sensitive to that, whereas other individuals seem less sensitive to that perturbation, blocking estradiol. So we decided to take this approach in the clutch size thing and see if that would give us a unique insight. We did this in zebra finches. The nice thing about zebra finches is you can get them to lay extra eggs if you take eggs away as they lay them. And so we used an egg removal protocol here. And then what I plotted is a reaction norm of a first breeding attempt, which is a control for everybody, and then a second breeding attempt where we had a control group again, and then for the other group, we actually took eggs away. And what you can see is we massively manipulated the phenotypic trait. So we're extending that phenotypic variation. The idea being that will allow us to identify a physiological mechanism a little bit easier. So here's a reaction norm for the clutch size itself. Here's the reaction norm for prolactin, and you can see it's a dog's breakfast. So this is the first nail in the coffin for prolactin, as far as I'm concerned. You can see lots of variation. That might be interesting. I'm pretty sure it is an assay variation. I worked with Pete Schock on this, who's the expert on prolactin at the time, so I trust the data. But when you look at the relationship between those, the association between those, there's absolutely no relationship in individual females between the change in prolactin and the change in clutch size. So we're starting to uncouple this idea that prolactin has anything to do with that. But with our reaction norm approach, because we got birds laying up to 17 eggs, we could actually blood sample them 3 times. And again, zebra finch is incredibly tolerant. And so we now have a reaction norm approach to this where we can look at the slope and the elevation. And we were interested to see if you look at a nuanced view of prolactin, do you suddenly see a good mechanism? And the short answer is no. And you're probably wondering, why would you say that then? Well, I'm going to say it for 2 reasons. I don't want to go back. Okay, 2 reasons why I say that. The first is that I really don't think there's any evidence at all prolactin is involved in clutch size determination. And my bottom line was, if you want a knowledge gap that's worth investing your time on, this is one of them. It just blows me away that 50 years on, 60 years on, we cannot explain clutch size variation from a physiological perspective. Okay, I'm going to go through this quickly now. Even quicker. Deep breath. I'm just going to finish with 3 examples asking a more general question about whether reproduction is costly. And I'm going to start with whether egg production is costly. And some of this will have reaction norms in this. I'm going to reinforce some of these ideas, and this is all female specific. I've been guilty like everybody else. When I write my egg size paper, I start by saying, it is well known that egg size has fitness consequences. Well, I stopped doing that because that's embarrassing. But, you know, egg production is costly. It seems to me a no-brainer. And you get people's attention. They go, oh, yeah, egg size is costly. Well, I'm not sure it is. And so we started trying to address this question. And I actually think we're asking the wrong question again. I think we should ask how often and under what circumstances is egg production or reproduction costly. And I actually think that the answer to that is not often. I think most of us go into our studies thinking that we're bound to find a cost of reproduction, because of course reproduction is costly. And then we don't find it and we get kind of freaked. Well, that's weird. Why wasn't the trade-off there? If you read Stephen Stern's, and you should absolutely read Stephen Stern's life history book, 1992, there's one book you should read. It should be that one. He actually dealt with the idea of trade-offs and costs of reproduction and argued that they should be pretty rare. We shouldn't see them very often because they're his view was they're resource dependent. And in most cases, resources are not that critical in terms of the resources you need for breeding. So I'm going to give you hopefully 3 quick examples. I'm going to talk about energy management during egg production, reaction norm story. I'll talk about the physiological cost of reproduction, and I'll talk about parental care and a walk in the park. And it honestly gets quicker, so we're right for time. We started off actually asking the question, because my focus was on egg size, is egg production costly? And going into it, we kind of thought, well, females, non-breeding females aren't producing eggs, breeding females are. They're doing something that non-breeding females aren't doing. There must be an additive cost. And again, conventional wisdom is largely that things are additive, and I think that's wrong. So we wanted to test this idea. And we actually went with energetics, which for those of you who know me, I don't have much time for energetics. I'm not really sure it's a big currency of anything. But in this case, we tried to work with the system. So we measured RMR and DE, and we've got non-breeders on the left and then laying females on the right. And what you can see is we got about an 18% increase in RMR on average, but we got no significant increase in DEE, daily energy expenditure, which is a little bit weird if they're doing something additively, particularly costly, producing eggs that they weren't doing previously. What got our attention was the range of variation around those laying birds. So DE actually decreased. We did this in individuals again, so repeated measures. DE actually decreased by 33% in some individuals and increased by 44%, 48%, 60% in other individuals. And then we asked, well, okay, this could be an energetic challenge, but how good are zebra finches at dealing with energetic challenges? And basically, this is a bunch of repeated measures, and what we've got here is thermoregulating and adapting individuals. So we actually challenge these birds at 7 degrees and then either measured them at a warmer temperature or not. So we've got an increasing demand here, and you can see this is a separate study, but we confirmed that change in RMR of about 24%. But if you look at it relative to these numbers here, it's trivial. Yeah? The cost of egg production is trivial relative to what they're able to do. And what's amazing to me is we've actually got laying birds here. So these are the ones that we've acclimated to the temperatures and then measured them at the higher temperature. But here we've actually got thermoregulating birds laying eggs at 7 degrees C, and they're able to do it. And what you can see intriguingly is the laying birds actually decrease their energy expenditure, which is really odd. And so, this reaction norm approach gives you a different perspective on things. And I think it means that detecting energy costs of egg production, if they are there, is really complicated. We didn't leave it there. We kept measuring the same individuals again and again and again. And so what this shows is actually the change in RMR relative to the change in DE. And this shows you what I call energy management. And I don't think anybody, Bob Rickliffs wrote an article on this, and people didn't pay enough attention. We all get a single point estimate of energy expenditure, DE or RMR, and we think we can interpret it, and you can't interpret it. So what we see here is some individuals are able, apparently, to decrease their RMR enough that they either don't increase DE or even decrease DE. And then other individuals do have additive effects. And then we went really crazy and did this whole thing again. So now we've got 4 repeated measures. And I understand this is asking a lot, and probably nobody's ever going to do this. But we find repeatability of energy management. So if you look at how they change their DE, do they decrease it or increase it? That's repeatable across breeding attempts in individuals. To me, that's huge. And it kind of suggests that individual variability in energy management, confounds interpretation of point estimates. And just very quickly, a couple of components of that. How do they do it? They change locomotor activity massively. And we now know that our starlings do this too. Laying females just aren't as active as non-laying females are. So that's one thing they do. And then they actually change food intake. And so not surprisingly, when you have a non-breeder and you thermally challenge them, they eat a large amount of food. But when you actually ask them to lay, they actually decrease their food intake, which is really weird again. And so, if we're working on these sorts of issues, we've got to take this stuff on board. Physiological basis of cost of reproduction. So get me to my last current work, actually, which I'm very excited about. It's a shame it's a last year event, but there we go. I wanted to see, so backstory here is the very early work on cost of reproduction by evolutionary biologists argued that that was a physiological thing. Okay, so cost of reproduction is a physiological trait. And so what frustrates me is trying to get my fellow endocrinologists and physiologists on board with that. There are huge opportunities for us to take life history variation and dissect the physiology, not because we think it's important, because evolutionary biologists think it's important. And so we tried to tackle here the cost of reproduction. This is our field study system. So we do experiments. In this case, we wing clip to manipulate workload in theory, because they're flying around. We get all of the standard reproductive metrics. We blood sample them. And importantly here, rather than just measuring one thing, we measured 13 different traits, which is a lot of work, but I think you have to embrace the complexity of physiology. You can't just measure one thing. And then we did radio tracking, initially handheld, but now we've got an automated radio tracking system. And so we can get these samples, and look at physiology in relation to current reproduction. But then in starlings, we can actually do, they do a second brood, so we can look at future fecundity and technically survival. And then they come back in year 2. So we're pretty close to fitness here, I think. These things only live for about 2 years on average. So if we get lifetime reproduction in 2 years, then we're doing pretty well. And so here's the results for the first year. We got, so the important ones to focus on are the clip RT. So basically controls, if you put a radio on, which you have to do if you're going to do radio tracking, that's already a challenge, handicap. And then we had clipping only with no radio. And then the final group is the doubly challenged ones. We wing clip them and put a radio on. And what you can see is that's the group where you push them far enough, they have lower current productivity. They pay a future cost. So in terms of the proportion initiating a second clutch, those clip RT birds didn't bother to breed again. And then this is not significant, small sample size and tough, but quite a substantial decrease in survival. So we have a manipulation which has generated a life history cost of reproduction. That's good. Now we look at the physiology. And when we looked at our 13 traits, 7 of them fell out as being different in that clip RT group. Hematocrit, hemoglobin, uric acid, NEFA, triglyceride, haptoglobin were lower, and then higher DROMs. And amazingly, best result I've ever had, when you fed those into a PCA, you get this fantastic linear relationship now, where you even in the clip-only birds, you start to see some evidence of a decrease in physiological state or condition. And that's only when you push birds really hard. And so, yeah, 5 minutes, we're good. So here's the problem. Then I repeated the whole thing, which is a good message. So if you want a message, always repeat your work. And the first time I did this, I thought, oh my God. So this is the result from the first year. Did it again, absolutely nothing. Same treatment, different year, same physiology, completely flat line. And so I started to think about it because that forces you to stop and go, okay, I thought I understood what's going on, but I don't. And so that led to a review I wrote, and it led to my current insert proposal, which I'm just going to finish with. So how do females escape a cost of reproduction? Back to this energy management kind of thing. We get a difference in the use of adaptive mass loss, which I think it is. Mass loss is not stress, it's adaptive in this case. They did that to avoid the cost. They actually decreased their activity by about 22%, which is really weird if you have this conventional wisdom view that parental care is working as hard as you can. Going, flying off, getting food, coming back, flying off, getting food. There should be no flexibility in there for you to suddenly say, I'm going to work 20% less hard, unless it comes at a cost, and it doesn't come at a cost. So even though they decreased their activity by 22%, their productivity was exactly the same. So once again, we have a complex world out there. And this led me to start questioning how hard birds actually work in general. And I'm going to finish it. And it's just it's our future vision. I got into this again just with natural history. So I went to my starlings and said, okay, what are you doing during your day? So it turns out they take about, out of an hour, they take about 2.7 minutes to feed their chicks. We know their flight speed and flight distances from our tracking, and they probably spend about 10 minutes or perhaps even half of that to actually fly backwards and forwards. And so I have no idea what they're doing the rest of the time. It looks to me like they've got 78% of their time budget totally unaccounted for. And probably a lot of that is foraging, but I can't see that as being high intensity. And this goes back to these original ideas that the cost of reproduction comes from sustained high intensity activity. Okay? So this is Grant and Dahl's original idea of this 4 times BMR, that animals are all working at that level. Their assumption inherently is that every individual is by and large doing that, which we know isn't true. And they can't do any more than that. And if they work that hard during parental care, they have to pay costs. Okay? And so we're questioning that at the moment. And the approach we're taking is, where does the cost come from? Where could the cost come from? So flapping flight, 9.2 times BMR, without doubt a high intensity activity. Is it sustained? Do they do a lot of it? So my European starlings fly for about 18% of the active day. A whole bunch of other birds seem to fly even less. So they're really not spending a lot of time in the sustained high intensity activity. Every time I give this talk, someone says, well, what about aerial insectivores? Maybe, but then this fantastic paper came out showing that swifts basically harvest the energy, the environmental energy, and get a lunch for free. They get so much energy from gliding that it costs them absolutely nothing to forage. So again, I think we just got the world the wrong way, and this is my, I think, final slide. We're exploring the ecology of inactivity. This isn't me first. Several other people have used this, or they've used the ecology of laziness. But if you hear it later on, I heard it there first. So we did a quick web study, pulled out a bunch of data. Ducks, for some reason, people like doing time activity budgets on ducks. We measured these activities, and we're really interested in inactivity versus activity. And just a glimpse at some of the results. This is my PhD student's work, so I told him I wouldn't give you too much information. I'm desperate for him to publish this before we get scooped. What you can see is that on average, across a whole bunch of different life stages, animals are inactive for about 40% of the time. And if you look at the flight thing, thinking again that if Brent and Dana are right, the flight has to be highly sustained activity. This is the graph here. Flight's down here at about 6% across all our taxa. And foraging's, a lot of time, but a lot of this foraging doesn't look like it's particularly energy intensive. Yeah? And if you look by the side here, resting or perching is what most animals do to spend most of their time. So I'm just going to wrap up. This is my mantra thing here. I'll go right to the end and just explain this to you. This is for the graduate students in the lab or the other faculty. Don't take the road most traveled. It's really easy. And I know I've preached this to graduate students in my lifetime, and they go, well, we can't afford to take the risks that you could as a tenured professor. And I agree with that. But just doing confirmatory work isn't going to push the field. Read widely. None of you read enough. I know it's hard, and you don't read old stuff enough. But you should, because but some of that was good. A lot of it wasn't good, but some of it was good. And knowing it and building on it rather than just repeating it and then appearing not to know about it is really bad. People take big answer big unresolved questions, and I've given you a bunch of them. Physiological mechanisms of life history traits and products in general. Always question conventional wisdom. Most people think they know the way the world works. They're all wrong. And persist and don't give up, because it'll be worth it. Okay, thank you very much. Sorry that went a bit long, but yeah, thank you.
I don't try to get more of it, which I really apologize.
No worries.
I'm used to it. If somebody has a good idea how to elevate average to the next level, I've never seen the term, but we would like to be the first.
I think getting people back in the room is the objective. Unashamedly so.
Let's do that. All right, perfect. So we have a bunch of people in the room. Are you monitoring the online chat? If there's any questions, let us know. So questions from the audience?
Yeah.
Is there anybody?
No, I'll
see. So this is great, this cost of reproduction thing, and assuming cost. So Andy and I, way back when, looked at the energetic cost of growing because it looks like it should be very energetic.
Yeah.
Physically they're moving loud sounds. No more than the cost of put it around too. So they're in a totally different system. Working with assumptions that things are a particular way because we just all talk to each other like we should. So that was fun to see.
So just to comment on that, we went through this idea that singing is an important thing and costly thing, and I thought there was really good evidence it really isn't. And then I looked at a wildlife documentary last week, and it's got a singing bird, and it says it's well established that singing is a huge cost for male birds. Don't stop it,
Yeah. Well, I mean, even if we look at the energetic cost of bacon, the same thing.
Yeah.
It looks like it
Yeah.
should be cost, right? All that. No. So anyway, I just it's neat to see that you're coming at things. I had piles of questions. I'm just gonna ask.
We're gonna have lunch, so we're good.
Yeah, right. So my one little question is in the trade-off reductions to the emails of motor behavior decrease with no effect on life. Is there any evidence that males compensate on?
Well, so in our zebra species, we haven't done that because we're female biased. I refuse to. I use those as an excuse to explain my female. But the answer, as you know, when people have done brood cycle manipulation, things are handicapped, there is some compensation. we didn't find much in our starling study here. The males didn't compensate enough, for sure. And it was actually a pretty, but our males don't work very hard in the starling system. But I think in some cases people have found males compensating, but usually not 100%. So it's quite common that males respond to that, but usually not enough to make up the parental care part, but then you often don't see costs.
And just for the online audience, if you could ask questions via chat, we have some background noise here. That's why you guys are muted. So please ask questions via chat if you can.
Fred, your first question in the last three that you discussed here is egg production costly. I was just curious with the line that you use, it's trivial compared to what they can do in terms of when you stress them out in the environment for these goals and such. Because I mean, say I'm doing this is obviously a human comparison, so very different. But if I'm working on an assignment and I say up to 48 hours, I can do it. But if I keep doing that over time, it's going to have a huge cost in terms of myself and also how well my assignments turn out. So maybe I just missed this, but how long, like how long was the sampling that you were doing with the egg production over how many years with the same females?
I'm not sure I totally understand. I mean, basically, I've thought about that. So egg production takes about 10 days from the start of it to the last egg. And it could be that females just absorb that debt and pay it off later. And in my penguin systems, for example, the female does a lot of the, she does the egg laying, not surprisingly. She then takes the male takes the first shift because at the end of that, she has to go to sleep. So without doubt, I think there's some deferred costs there in that system. But I think the argument was that if you just look at it from purely energetic point of view, so I'm not saying there might not be other costs, and I actually think there could be physiological costs which are not energy dependent. I kind of have been pursuing this non-resource based trade-off idea that there are complex physiologies out there that are incompatible. In the penguin world, it's migration and reproduction. Those two things just don't work together. And the birds are migrating back at full speed to the colony, and that impacts their egg production. So I just think that in terms of the energetics, which is what we're looking here, what is the energetic cost? If it's that plus 6% or even that plus 24% relative to what they can do. And okay, so your question was how long was that? It takes quite a while to acclimate them and then get them to breed. So they're doing the same thing. They're doing about 10 days at low temperatures, having been acclimated or not. Yeah. So yeah, I just think that the point of that was that they're able to upregulate energy expenditure a lot more. And actually they can do that and still produce eggs. So those birds at the top, they were producing eggs, blew me away. I mean, the fact they can lay eggs at 7 degrees. I mean, they are desert birds in Australia. So
another question, a couple of rows back.
So you try to manipulate the hormone signal, and you said that the receptors are limiting how much they can pull in. You try to think of a way that you could manipulate the promoters for those receptors to have the birds actually generate extra receptors so that they can put more.
This is why I give these talks now, because I'm too old for that. Like, I'm actually going to propose an RNA-seq approach for the clutch size thing, because I think we need a fishing expedition there. I hate fishing expeditions in general, but if it isn't prolactin, someone needs to identify what it could be. And so I think if we can, use zebra finches and the flexibility to sample just before that inhibitory signal and after, and then do RNA-seq and look for it. Otherwise, I just think I'm too old for that sort of stuff. Like, I don't want to tool my lab up. We've never really gone that way. I'll be honest, I'm not sure that just genomics is necessarily the approach. Yeah, I mean, that Marcel Visser's stuff, we looked at every gene and every genome, and there's no strong signal from anything. The power of that is inherently bad. We did a GWAS in theory. We actually started by selecting for early and late laying. Perfect system in theory, and he could breed them in captivity. And the idea was you do a GWAS and suddenly certain genes for early laying versus late laying drop out. Well, they didn't. There was nothing. The only things that were of any interest were LH receptor genes, for example, which confirmed, what we thought was where the block is. I think the worrying thing about that is I don't think anybody's going to get money to do that again now.
Yeah, I'm just thinking from a coffee perspective. So, like, if you start drinking coffee, you build more receptors for caffeine in your brain.
Yeah.
So like, could you get follicles or like the tissues that receive follicles to build more receptors?
Yeah.
But you have to start earlier in the process, like you think that's something.
Yeah. So it's a great question, but it really addresses this issue of if we're talking about this as the basis of a trade-off and as a marker of individual quality, then I agree with you. It's a fascinating question about how do you end up with more or less, and can you do anything to improve the number of receptors if that really is the limiting step? But it's just people don't think the world works that way. Yeah. So I don't really know what to say to you to say, well, how could that happen? But I think focusing on that would at least get us looking in the right place. And then hopefully a clever person will come along and take it on.
I had a quick question as a complete, I know nothing about these processes. So the variation in egg size you started with, is that related to the order in which these eggs are laid?
That's the big thing. So intra-clutch, within clutch egg size variation, average is about 3% in passerines. It's not very much at all.
Egg size.
Yeah, but this is the weird thing. 3%, everybody got very excited about that. In the 70s and 80s, everybody was looking at intra-clutch egg size variation, trying to work out fitness consequences. Why does egg size decrease towards the end? Why does it increase towards the end? And it's all fascinating, but it's missing the boat. If you're interested in egg size variation, you're missing the boat. You're looking at that small amount up there, whereas you should be looking at this. So that was part of the reason. And I mean, I got the extreme is the macaroni penguin system, where they lay they only lay 2 eggs, and they lay a really small one and a big one, which is about 60% bigger. They're the world record holders for intra-clutch egg size variation. That first one never survives, never produces a chick. So there's a fascinating question there. But by and large, I think we are throwing a huge amount of effort at the wrong question, or at least a less important question than the action. If you've got your eye on variation, look at the big thing, not the little thing. And that's what's happening at the moment.
The online audience, any questions? No? And yeah, you had one last question.
I was just curious, what have been changes, if any, within the animal ethics approval in terms of getting studies done throughout all the years? Since I mean, you are full of hormones as well.
That stuff's relatively easy because most of these effects are benign, relatively benign. Like there's no point in doing something that stops my birds breeding, because they would be pointless. And even in the starlings, we need the birds to come back the next year if we're going to do future fecundity. The harder part, I'll be honest, the data I showed you on changes in oviducts and stuff, it required us to kill a large number of females. I don't like that, but I needed to do it that way, and it was easier to do back then. I don't think I'd get permission for that. The only thing I would say in my defense is when we killed those females, we made as much use of them as possible. Collaborated with all sorts of people to make sure that maximize the value. But it is becoming an issue that, I mean, I work very well with my animal care committee. We're very diligent and they appreciate the fact I fill out forms on time and simple things like that. So I can still get permission for this stuff, but I suspect if I wrote to them and said, I want to kill 100 birds, they could say no. Yeah, it's interesting. And my sense of the UK, which is where I'm from, a lot of the field physiology and endocrinology was being done is not being done. People just have given up the fight because it's so hard to get permission for any of that.
It's interesting. I was at a conference just last week where this issue was discussed in the realm of different human-animal interactions more broadly. And there were a few people who said that they initially resented the additional hoops to jump through, but it changed the culture in a way that they feel is a better culture.
Yeah.
And they didn't feel it did make them ask less important questions, just ask them a little differently. So I thought that was an interesting comment.
Yeah, I think there's nothing wrong with forcing us to think really hard about it.
All right. Well, let's thank Tony very much. We'll see you around for a couple more days.
I'm around today. I've got a chairs meeting starting tonight, but yes, thanks.
So if anyone wants to
Today would be better.
with Tony, maybe a function back.
Otherwise, I gather you have breweries downtown. So maybe there.
No.
We can do that.